Welcome back to my YouTube channel. Today we're going to be talking about three things. We're talking about light, vision, and movement. The way I'm going to structure the video is first I'm going to talk about what is it not going to be on, and then I'm going to tell you about what it is going to be on in a very general sense. And then lastly, the biggest part of the video, of course, is going to be me going over everything in detail. First, what is this video not going to be over? So it is somewhat implied that these two things will be gone over, but I'll just clearly state them here that they will not be, which is that we're not going to be talking about the specific individual neurons firing, and we're not going to be talking about the specific muscle fibers contracting. Those are the two things we're not going to talk about. What we are going to talk about, however, is all the neurological areas in the brain communicating with each other in a very structured format to either, in the case of vision, produce a conscious image over time by taking in a bunch of raw features from the eye, and then increasingly make them more complex, analyzing this part, analyzing that part relative to each other. Or in the case of movement, the neurological areas associated with creating a goal, making a muscle sequence around that goal, and then executing that goal. And then while you execute it, you're having some sort of feedback loop in which you fix any incorrections. Okay, so that's not what we're going to be. Those are the things we're not going to talk about. First, before I go into detail, I'm going to go over this entire flowchart in general in a very general format. First, before I do that, I want to say that everything is color-coded. I feel as though that's kind of implied based off of the different colors. And what's also implied by that is the fact that if something is in red, everything kind of connects to everything. One more thing that we're not going to be talking about is specifically how vision correlates to movement. We will be touching on it, but we won't be going into it in very detail because when we think about motor movement, there is a lot of nuance, a lot of niche factors that play into creating the goal in the first place. But once we have that goal of the movement plan in the first place, then that's where we're going to talk about what happens after. But we won't really be talking too much about how you come up with a goal in the first place. And the reason for that is because that's very complicated, that's very nuanced. That has to do with a lot of higher level thinking. And higher level thinking like that, there's just too much to cover, so we're not going to go over it. We're going to mention some things. So first, let's talk about everything in general. So as you can see right here, light is produced by some sort of source. These three incandescence, luminous, and synchrotron radiation. Also, one more thing I want to say before I go further is that a lot of this stuff I've never heard spoken in person. I've only read about it. So if I mispronounce something, that's probably why. So let's go over this. So light is produced by some sort of photo source. It travels through air, and then it travels through the anatomical structure of the eye in such a way so as to cause it to refract and bend at certain angles until it hits the back of your eye. Once it hits your back eye, it's going to hit two things, cones and rods. Cones respond to color, and rods respond to light, either bright light, dim light. That's the distinction between the two. For the sake of consistency, we're going to talk about this more so in terms of rods than cones. So let's do that. So everything in purple has to do more so with a phototransduction pathway occurring within each individual rod, which is just basically a very fancy way of saying that within these rods, in order to respond to light and produce a neuron active potential, like neuron firing, that sort of thing, you have to have this really long and somewhat complicated this really long pathway, this process whereby this thing affects this thing, by this thing affects that thing, and that's the sort of the idea by it. So yeah, and then it all starts with a chemical called retinol, which is sensitive to light, such that when it gets hit by light, it absorbs the light, and by absorbing it, it changes shape, and that's kind of the beginning of all this. It changes shape, it causes something else to change shape, and then it does this to this to this to this and this. That's the idea of what's in purple. It's just this thing affects this thing. And ultimately the idea is that the rod will be activated. Once it becomes activated, it does not send a neuron, it's not a nerve itself. It doesn't send an action potential itself. What it does do is it influences and activates a bipolar cell, and that bipolar cell will, if it chooses to, activate what's called a retinal ganglion cell. And then the retinal ganglion cell will send its action potential impulse by its axon through the optic fiber until you get to the lower and the higher level brain areas related to vision, which is what we have here in dark green, blue, whatever color this is. I don't really care what color you call it, it's not important. But that's the basic idea of this. So light's produced, travels through the air, goes through your eye, is turned into an electrical impulse. That electrical impulse now goes through the brain. So the basic general overview idea of how all of these many different signals from the eye are sending to the brain, how the brain in its various areas interprets the signals as makes meaningful information out of it. Very general overview idea of that is the fact that we have all of these little areas. So the entire, or not the entire, so the primary visual cortex is located in the occipital lobe, which is in the back bottom of the brain right here. It kind of wraps around in succeeding fashion such that visual area 1 is the bottom and then visual area 2 is above it, and so on and so forth. The idea is that as you go from visual area 1 to 2 to 3 to 4 to 5 and so on and forth in a higher fashion like that, raw features from the eye about what is being seen becomes increasingly interpreted and increasingly more complex to form and to interpret more complex features relating to the vision, to the visual image that is being seen. That's kind of the idea. There's some more niche aspect to it, which has to do with, well, I won't go into it, about prediction, but we'll talk about that in a minute. So that is everything so far. We have light's produced, travels through air, travels to the eye, is turned into an electrical impulse by way of a phototransaction pathway, goes to the brain. Brain has this sequence of different areas dedicated to interpreting different raw features of what's being seen. And then ultimately have an image that is being produced as a sort of static, dynamic product, not like a stable, not like something that's just like, we'll talk about later. So that's everything thus far. Now let's talk about movement in a very general sense. So the idea of movement is that kind of like the visual areas of the brain, we have different areas relating to movement. And the way it works is ultimately what we want to do is we start out with a goal, and we also take in, and the goal comes from higher level cognitive thinking, which again, there's a lot that goes into that, which is why we're not going to talk too much about it. And this goal is converted into a movement plan that is converted into a movement plan with specific muscle sequences firing in specific times and with specific intensities over time. Taking into account also external cues such as where you are in space and where that thing is in relative to you and how much effort would be needed, and sort of like environmental cues like that. So again, movement is all about creating a plan, refining that plan, so making it better, and then executing that plan. And as you execute it, you actually will further refine it as you're doing it, and to correct any mistakes that you make along the way. Another general idea over this that I guess I'll point out is also this idea of affordances. I don't know how well you can read all of this. I hope it's pretty eligible. But affordances is kind of just a fancy way of saying that a big idea behind this sort of motor movement planning pathway is that what actually happens is not like as if you are in the brain, it's not like as if the brain is creating one singular movement plan at a time in relation to its surroundings and then deciding to go with that plan or not going with that plan. What's actually happening is that at any given moment, while you're taking in visual stimuli or somatosensory stimuli in general, you're actually creating many plans of possible, and many possibilities, many possible plans that you could carry out and execute, called affordances. And that's where we use affordances to describe the fact that there are many plans going on that you're creating. But of course, among all these many different plans, you can't all carry them all out at once. There has to be only one at any given moment. And so one of the ideas behind movement is that most of these movement plans are inhibited and basically said no to in a way. And it is usually, and it's really typically what you see is that the only movement plan that you actually end up doing is the one that you weren't said no to. That's kind of like a general idea behind that. So, yeah, I think that's a pretty good overview of everything. Now let's talk about everything more specific to detail. Okay, I think it's good. So, okay, let's start off over here. So, of course, everything in red is light. This first half of it everything to the left of this pin has to do with actually light itself, and then everything to the right of it has to do with light traveling through the anatomical structures of the eye. So I suppose really the way I should have worded this, the color coding, is I should have kept this all in light and red, and then I would make this eyeball, and then this would be photoreceptor. That would probably have been more clear way of going about it, but it doesn't matter. So these are the three ways by which light can be produced. These are the sources of light. There are other subtypes relating to these three main types. However, that's not really, my goal isn't really to get into the specific niche parts about that. I just want to talk about the three main ways. Again, don't know how to pronounce these words because I've only read it, never heard of it. We have incandescence, luminescence, and synchronous radiation. These two are the more common ones you see. Synchronous radiation is a very niche form involving a particle accelerator in which charged particles, usually electrons, are traveling in a curved pathway that it normally wouldn't be traveling because there are magnetic fields placed in the particle accelerator to cause it to do that. And through the course of it traveling in this curved pathway, it somehow emits electromagnetic radiation in the form of photons, visible photons. Again, don't really worry about this one. This is just something that I thought maybe it's worth mentioning. And then we have incandescence and luminescence. Incandescence has to do with heat, and that's why I wrote the little triangle here. This is a delta symbol. It symbolizes heat, specifically in chemical reactions when we're writing them out on a piece of paper. What happens with incandescence, how we produce light, visible light from it, is that atoms within some sort of piece of matter are vibrating, and through the course of them vibrating at a high enough amount, like its kinetic energy is high enough, it actually will emit photons, visible electromagnetic visible photons as a result of that. So that's one way by which we can do that. The next type is luminescence. Before I talk about this, I want to point out the fact that energy cannot be created or destroyed, it can be converted to forms. And the idea with luminescence is that you have an atom, okay? You have a nuclei, and the nucleus, which is the protons and the neutrons, and the electrons that are surrounding it, going in all these weird movement, these weird patterns of movement of probability called electron clouds, and we have quantum numbers that describe that too, but that's not really the point. So with luminescence, electrons are normally in their ground state. I don't know how you can read this. This little area going up represents energy, so higher energy, the higher you go. There's this little slot here and this little slot here. This slot on the bottom has lower energy because it's lower on this little energy diagram. This is the ground state. Most electrons, whenever they're just being normal, they are in their ground state and have less energy. When they are excited, however, for whatever reason, they will move up into a higher slot within the electron cloud pattern that they usually follow, and they are in what you would call an excited state. And the idea behind how this whole process produces a photon is that when it goes back down from the excited to the ground, so it's going from higher energy to lower energy, it will emit energy in the form of a photon. Because again, like I said before, and the reason why I said it is energy not can be destroyed, it can only convert forms. And that's basically how you get that. You absorb energy from some way, don't really care, and then you let go of that energy as you move back down to a ground state in the form of a photon. So yeah, it's the first part. So what is a photon? Photons are massless, discrete packets of pure quantized energy, like pure electromagnetic energy propagating through space. If it's propagating through a vacuum, meaning there's absolutely nothing there in space, it moves at the speed of light. A few other things relating to photons. A photon is a visible light, is photons, and it is part of a larger spectrum called the electromagnetic spectrum, which has to do with the fact that these pure energy moving through space, oscillating through space, kind of like this, when it oscillates, it goes up and down in predictable ways, such that the part in the middle before it goes up and down, that part in the middle is the equilibrium position, and then the apex is either side where it's up or down. Wavelength is the measure of distance between either apex, and frequency is how many times it oscillates in a given time frame. So general idea, you have the electromagnetic spectrum. Visible light is just a small part of that spectrum, And the way that we say where it is on the spectrum, the way we quantify that is based off of its wavelength, which, like I said before, photons exhibit wave-particle-like duality, meaning that they can either behave as a particle or as a wave in the right given scenario, which was shown through the slit experiment, where basically by measuring by measuring a subatomic, by measuring it, something about it, this measuring is not a passive thing, it's an active thing, and it's influencing and causing it to change behavior from wave to particle, particle to wave. So again, photon visible light is just a part of a much larger visible electromagnetic spectrum, and it's distinguished by its wavelength. And as you can see in here, I wrote here 380 to 750 nanometers. This is the range. And also another thing about the wavelength also determines what color you would perceive with 380. Another thing, so higher frequency equals lower wavelength, and higher wavelength equals lower frequency. Blue light is a lower wavelength, therefore higher frequency, therefore higher energy. And then red light is on the other side of that, in the sense that its wavelength is longer, therefore its energy is less, and its frequency is also less. As you can see, I wrote up here, E equals HF. What does E equals HF means? E equals HF means energy equals Planck's constant, which is the H, times frequency, which is the F. And this relates to how you determine the energy of a photon as a visible light. Okay, so now we have talked about the source of energy, or the source of light. We talked about a little bit about what photons, visible light really is. Now let's talk about how photons propagate themselves through space, ultimately leading to your eyeball. So photons move through air mostly. They can do a number of things. They can be absorbed, they can be refracted, such as if it goes through glass, they can bounce off of it and then bounce off to something else, until eventually, by coincidence, it reaches your eye. Now, all of these sequential little steps has to do with the anatomical structures of the eye through which it passes in order to be refracted at specific favorable angles to land at a specific favorable part of the eye, or the back of the eye. One thing I want to point is that whenever you look at an object, it kind of actually, because of the way it refracts, the top of the object and the bottom of the object kind of like flip because of the way that they enter the eye and refract. So normally, if it wasn't refracted, the brain will basically fix that and change that whenever you have conscious vision. You would actually see everything upside down. But I guess that's worth mentioning. So two things I want to mention. The cornea and the lens are the two important, significant anatomical features of the eye that will actually be doing most of the refracting. Now, the difference between the cornea and the lens is that the cornea will do more refracting than the lens will, and that the cornea it can't really change at any given moment that amount that it does. It's kind of like a baseline amount, and it's kind of always there. The idea behind that is that the lens will basically, the lens will make up for whatever amount of discretion it needs to change based off if the object is closer or farther away, by either becoming more curved or more flat. And it does that by using the ciliary muscle that it's attached to. So yeah, so let's talk about this. So light's produced, travels through space, just so happens to travel to your eye. Once it goes through your cornea, it's refracts some, then it hinders through the anterior chamber. Anterior chamber has aqueous humor, and I guess we'll talk about this later. The vitreous body has vitreous humor. So it goes to this like liquid-like substance, then it goes to the pupil, then it goes to the lens. For the lens, if something is near or distant, again, like I said before, it will change its behavior. It changes its curvature, whether it's more curved, more flat, depending on whether it's near or distant. So if it's near, there needs to be more refraction. If it's far, it needs to be less refraction. And that has to do with the fact that if something is far away, what actually ends up happening is the light rays are somewhat more parallel as compared to if it was closer by, it would be much more at an angle. And what you want is to be more parallel. So then it goes to the lens kind of refracts it, however, based off of something's closer enough, something far away. Then it goes to the vitreous body that has vitreous humor. So I guess if we wanted to compare and contrast the aqueous humor and the vitreous humor, some of the things that we could say is that, hence the name, aqueous humor is more water-like, and the vitreous body is more gel-like. That just has to do with the consistency of what that actually is. Aqueous humor, another distinction we make between the two of them, is that aqueous humor has a higher turnover rate than the vitreous humor. Basically what I'm saying by that is that I mean, you create more of it at any given moment, and you're also excreting, like removing some of it. So that's what we mean by higher turnover rate. The aqueous humor has more so to do with a few different functions, such as waste removal, nutrient delivery, and pressure regulation. By pressure regulation, I mean specifically intraocular pressure rate regulation. The vitreous body the vitreous humor within the vitreous body has more so to do with structural support, because it is also bigger if you look at an actual picture of the eye. And one more thing I want to point out, I guess a sort of conclusion we can draw from this, is that mostly whenever we're thinking about vision, we usually think about it in terms of all the different visual areas in the brain. This lobe community with this lobe, this area community with this area. And that is what it is mostly, I suppose, if you wanted to make that sort of conclusion. But the conclusion I want to make here behind what we're talking about right now is the fact that while it is, a lot of it is neurons communicating with each other to make meaningful information behind just pure electrical spikes, pure electrical signals, it is not just neurons. Vision has to do also with physiological support systems such as these. And if these go wrong, if there's some sort of pathology behind them, then you also have pathology just in general. So pathology here results in pathology in general, of course, and pathology basically anywhere else also results in pathology. So don't just think, oh, it's only just about the nerve firing, and it's also about the eyeball itself. Okay, so light's produced, travels to the air, travels to the eye, hits the back of the eye, the retina. The retina, like I said before, has either two types of photoreceptors. Photoreceptors is basically the functional unit by which light can be captured and through the process of phototranslation pathway, converted into meaningful electrical symbols, which gain more meaning and more value as it is transmitted to the brain areas, and the brain areas basically takes all this raw input and makes sense of it. So yeah, the functional unit. The distinction between rods and cones is that cones will react to and are sensitive to and process light of specific wavelengths. In other words, light of different colors, blue, green, red, and all of the other colors in between being a result of a blending of those. And for the most part, the phototranslation pathway for both cones and rods are basically the same in the sense that they follow the same logic. The distinction between the two of them in the phototranslation pathway alone is the fact that cones have a different starting molecule called phosphosphene. And with phosphosphene, like I said, you have the three types relating to blue, green, and red, different wavelengths. But for the sake of consistency and for the sake of time, I suppose, I'm only going to be talking about this term in rods, not really cones, even if they are pretty similar. So yeah, another distinction we can make between rods and cones that don't have to do with the phototranslation pathway itself, but other things relating to its actual macro structure in general, is hence the name. Rods are more rod shaped, cones are more cone shaped. What we mean by that specifically is that rods are more longer and shorter, or longer and narrower, and cones are more shorter and flatter. So if you look at like above of it, like a picture of it above, you would see that the rods are, something like this, and the cones are a little more flatter and look bigger from above, but it has to do with the shape. Okay, so obviously there are 120 million roughly rods and 6 million cones, which means there's more rods and cones by a decent amount. Rods fire slowly, cones fire fast. And another thing about cones is that you don't actually have equal amounts of red, green, and blue. You have more red than you have green, and more green than blue. And specifically, the exact proportions are somewhat like 60, 30, and 10, respectively. The reason for this isn't, I just want to point out, this isn't like completely random. It kind of seems like it, and it kind of seems, why would that be the case? But long story short, long and middle length wavelength is just more reliable, more useful for detail, and it's somewhat, in some weird way, it's somewhat cheaper for the brain to process under natural lighting conditions. And this is sort of like relates to evolutionary physiology, like what was the most useful, and whatever it is that is most useful to be used for survival, that ends up being what's more plentiful and what's more evolved and present. Don't really worry about that, though. That's not really something that I really want to worry about. Okay, before I get into the specifics of the phototranslation pathway, I want to point out one general idea, which is that, so this can be subdivided into two types, on and off bipolar cells. For the sake of clarity, we're going to talk about one, not the other. So in the specific type we're going to be talking about, the idea is that the rod, and again, we could talk about photoreceptors, rods, or cones, but for the sake of consistency, I'm going to talk about rods. So if I say rods, just think this could probably also apply to cones by way of the same general logic. Rods at any given moment are always turned on without light. So in the absence of light, the rod is on. The bipolar cell from which it connects to is always turned off in this scenario. When the phototransduction pathway completes because of light, it absorbs light, the opposite is true. So the rod will go from on to off, and the bipolar cell will go from off to on. Once the bipolar cell is on, it will then, whatever a retinal ganglion cell, which is the next part of the pathway that it connects to, it will influence that retinal ganglion cell such that the retinal ganglion cell might produce an action potential, and it's the retinal ganglion cell that actually produces the actual action potential that travels to the optic nerve into the brain. So the rods and the cones themselves don't actually produce the neuron firing, nor do really the bipolar cells. It's really more so about the retinal ganglion cell. Sometimes you might have a 1-to-1 ratio such that for each rod there might be one bipolar cell, and for each bipolar cell there might be one retinal ganglion cell. This isn't always the case. Sometimes you have multiple bipolar cells connecting to one retinal ganglion cell. One more thing I want to point out is that there is a very high acuity, high detail part of the eye called the macula, within that you have the fovea, where there's a very high concentration of cones, much more so than anything else. I don't know why I thought I should mention that, but I just thought I should. So yeah, another thing I want to point out before we go into the specifics of it, so that there's just more clarity about how this works in general, is the fact that part of what it means when I say that the rod is always on in the absence of light and the bipolar cell is always off, and to go from one to the other we have to absorb light, is in the absence of light, the rod is constantly influencing the cone and the rod is constantly influencing the bipolar cell and telling it to basically stay off by producing a neurotransmitter called glutamine. And then once we have the whole phototransduction pathway take place and go from step to step to step, what ends up happening is that the membrane will actually hyperpolarize as a result of the sodium and the calcium channels closing, such that this glutamine release is inhibited, it stops. So in other words, the rod is always telling the bipolar cell to shut up, be still, be quiet, and then once light hits it and does its thing, it basically stops telling it to shut up and be quiet, and will instead allow the bipolar cell to be turned on, and then, quote unquote, and then this turning on will influence the retinal ganglion cell. So what happens specifically? So within the rod, we have many discs. That's what these little horizontal lines are that I drew. And within these discs, you have a protein complex that starts with rhodopsin. That's what these little, this is what I got drawn here. So it has 7 subunits right here, so rhodopsin. Within these 7 subunits, you have retinol. Retinol in its natural state, in the absence of light, is in a cis conformation, which has to do with stereochemistry, just how it's arranged. When it absorbs light, when cis retinol absorbs light, it will, as a result of absorbing light, hence the HV, which is what that stands for, light, it will go from cis to trans. By going from cis to trans, it doesn't actually add any atoms. It isn't actually a real chemical reaction in the sense of it's adding or removing anything. It just simply changes shape. And by changing shape, by retinol changing shape within the rhodopsin, it actually influences the rhodopsin within which it is embedded to also change shape. And once it changes shape, it will influence other things around it. Specifically, it has 3 subunits called alpha, beta, and gamma. It influences primarily alpha. The alpha is the transducin guanosine diphosphate naturally. By changing shape, by the retinol changing shape and thereby changing the shape of rhodopsin, it actually influences transducin guanosine diphosphate to go from diphosphate to let go and have a triphosphate. So it basically removing something, adding something. That's sort of the idea by there as a result of change in shape. Once it does this, what actually happens is this alpha subunit, which is the transducin guanosine triphosphate now, disassociates. And when we say it disassociates, it is now activated because now it's going to go do something important. And the specific thing that I'm talking about that it does now that it is activated that is important is that it will influence another protein complex somewhere else on that same disc or another disc within that photoreceptor cell itself, which is specifically phosphodiesterase. So naturally speaking, phosphodiesterase is this enzyme that catalyzes a hydrolysis reaction by which Cyclic GMP go from that, by the way, cyclic GMP is basically hydrolyzed, meaning it breaks off into two smaller parts with the absorption of water, into GMP, which is guanosine monophosphate, which is what that stands for. So naturally speaking, phosphodiesterase is inactivated, just floating, just being on the disc, doing its own thing, or not doing anything. And once this alpha activated subunit disassociates from the rhodopsin, it actually travels to the phosphodiesterase and activates it by binding to it. And now this one singular active phosphodiesterase will actually catalyze the hydrolysis of many cyclic GMPs to GMPs. The idea is that as you do this, as there's now this transition from a higher concentration, or from a lower concentration of GMP to a higher concentration as a result of this hydrolysis reaction by the phosphodiesterase, you actually have what is naturally already present is the sodium and the calcium channels, which were already there within the rhodopsin, or within the rod cell itself. It will actually close, and by closing, the membrane is hyperpolarized, and then this is what we call when it is turned off. And by it being hyperpolarized and turned off, you also have glutamine release, like I said before, and is inhibited. And by doing that, like I said before, also the bipolar cell is quote unquote turned on, and the retinal ganglion cell is also influenced by a result of the bipolar cell being turned on. So one more thing I want to point out here is lateral inhibition and opponent process theory. Lateral inhibition is basically the idea is that we have another layer of complexity relating to this, whereby nearby adjacent rods or nearby adjacent retinal ganglion cells, when they're firing, if one fires and the other fires, they actually inhibit each other. And what that means, what the significance behind that is that this comes into play in opponent process theory. And opponent process theory is basically the idea is that the way that the first features that you notice at the beginning visual areas relating to vision of the eye, vision of the brain, the first basic features you notice is lines. And the lines that you most notice at first are the high contrast lines that contrast high, such that there is an area of dark and there is of light. Same kind of with color. Opponent process theory, the idea is that, again, the thing you first most notice is high contrast color and high contrast lines. And the way by which this happens specifically within the retina itself is this, again, lateral inhibition. That's the sort of the functionality behind it. Then the retinal ganglion cells, the axons attached to the retinal ganglion cells, they are propagating this action potential through these axons, eventually to lead into what we call the optic nerve. The optic nerve travels, its ultimate goal is to travel from the eye into the brain. Right here I have written in blue, green, whatever you want to call it, is the optic chiasm. Optic chiasm is basically this structural part by which the optic nerve fibers go a somewhat specific path from the eyeball to the brain. And it basically, at this optic chiasm, this anatomical structure, before it reaches the brain, it does this weird diverging pathway. A picture probably would be good for this, but I'm going to do my best to explain it with words. So imagine you have eyeball here, eyeball here, eyeball here. It wants to go to the brain right over here. Say that the nose is in between, so the nose is right here. And say the retina wraps around the back of it like this. The nasal side, say you divide it into half. So this half is nasal, this half is lateral. The nasal side of the fibers will cross over at the optic chiasm to that side of the brain. And then of this same eyeball, the lateral side do not cross over. And the same happens over here. That's kind of what we mean by the optic chiasm. And I don't know if that is explained well. Another thing that I wrote here, retinotopic mapping. Retinotopic mapping is the idea that there is an orderly, point-to-point, ordered fashion by which, how do I explain this? There's an ordered fashion, the point-to-point fashion by which certain areas on the retina are translated and their fibers run along to specific areas in the visual areas of the brain, so as to have a point-to-point sort of mapping out. So it's not like as if the fibers just go anywhere and it's just completely random. Retinotopic mapping is the idea that they have a very specific path that they follow from the retina, a specific part of the retina, to a specific part of the brain, and it's very specific. And in such a way so as that there's this pathology associated, such as if there's a lesion. If there's a lesion in the visual areas of the occipital lobe of the brain, there is a predictable and ordered specific part that you will, as a result of this lesion, that you will have like a blind spot in your visual field. I don't know if that makes sense. Same way if you were to like take a scissors and cut a specific part of the optic fiber, it will affect a specific part of this, like a local part. I don't know how well that was explained. So from the optic nerve, you go through the optic chiasm in order to reach the brain. So with the brain, you have two systems, the new system, the old system. The new system has to do with conscious vision, perception of an actual image. The old system doesn't have to do with an actual perception of an image. Instead, it has to do with these involuntary reflexes to visual stimuli that you don't really consciously perceive or think about. And another distinction is that most of what we're talking about vision, most of it has to do with the new system. And that's what I'm going to be talking about. The old system, not really worried about it. I just want you to know that it exists, I guess. The old system, an example of this, practical example of this would be that if someone has something wrong with the visual areas in their occipital lobe, such that they can't see a visual image, like a conscious perception vision. Everything else is fine though. What that means is that person is blind. But if you were to do some sort of specific like jump scare, like if you were to do some specific things to them, like flashing of light or something to that extent, they won't be able to consciously perceive it, but they will have a reflex reaction without even realizing why they had the reflex reaction. And that's to do with the fact that the old system, again, is not conscious perception, but rather instinctive reflexes. So don't worry about that anymore. Let's talk about the new system. The new system, so again, general idea of how this works. You go through the thalamus first, which is what the lateral genicular gyrus is. And then from there, again, like I said, there's all these different areas. All these different areas process increasingly complex and nuanced features of what is being seen in order to ultimately create a visual image. From V3, again, V3, V1, V2, V3, this is just visual area 1, visual area 2, visual area 3. So for the sake of time, we'll use the acronym and say V and then a number. V3 is a sort of branching part where you go from V4 or V5, and then you actually go out of the occipital lobe to different parts of the brain. And the basic idea is that information is processed from 1 to 2 to 3, and then for 3 we have somewhat of a branching, off-branching point from 4 to 5. And the distinction between these two sort of branching locations that we can go by, both are always happening at the same time. It's not like as if you only choose one or the other, is that V4 to the inferior temporal lobe, so like right here, has to do with what relating to the image. So what am I looking at? What is the pattern recognition? What is the significance of the meaning behind the object I'm looking at? Is it a face? Is it an object? What is it? And then the other one has to do with where. So this is more about spatial location in relation to the observer and in relation to you. Now, before I get into the specifics of it, which each sort of little visual area represents and means, I want to make a really, really important distinction. I want to say something really important. It has to do with this right here, prediction. So prediction process theory is the idea that, and I actually written here, I forgot about this. So prediction process theory is that unlike what you might be tempted to think, it is not you build an image and then after you build the image, you finish, you have the image, and then you predict what will happen next, and then you repeat this process. What it actually is that higher visual level areas in the brain predict both what will happen next, what will be seen next, and also predict what is coming in right now, what that will be. They make predictions, and then the lower visual areas receive these predictions from the higher level. They compare what is being seen, the raw input, with what is being predicted, and it only actually sends up to the higher visual areas, not everything it is receiving, but rather only the sort of mismatch error between what is being seen and what is being predicted to be seen by the higher visual level areas. This is what the adjust is, and it basically sends up to the higher visual area levels, and they adjust their predictions to create a more accurate prediction. And then it just makes another prediction. That's the idea, is that conscious perception of an image has less to do about processing everything that the eye sees and more to do about higher level areas making predictions about what is probably going to be seen, and then comparing, and the lower areas comparing that with what is actually being seen, such that they will only send up the errors. As a general idea, the brain doesn't directly sense reality, it infers reality. And also as another general idea, this whole thing is the the visual areas are not optimized so much as for accuracy as they are for survival. It just so happens that accuracy is very closely linked to survival. And that has to do with evolutionary physiology, because survival is what matters most. And so whenever we see that accuracy is such a big part of it, it's really more so a function of survival first and foremost. Yeah. So I think that's a good starting point. Let's get into the details. So the lateral genicular gyrus is as part of the thalamus. The thalamus is a very big relay station for everything that happens in the brain. So what happens is that, again, you have the optic nerve, crosses over, maybe it doesn't cross over, it's specific first to the optic chiasm, and then goes to the new system. The new system starts off with the lateral genicular gyrus, which is a part of the thalamus, which basically reroutes. It has 6 layers, which are distinguished by the parvocellular and the magnocellular. 4 of them are one and 2 of them are the other. I don't remember if 4 of those layers were dedicated to parvocellular and then 2 of them to magnocellular, or the other way around. I think it was the 4 of them were dedicated to parvocellular and 2 were magnocellular. Regardless, the parvocellular and magnocellular layers of the lateral genicular gyrus are basically 2 sort of different ways of processing the visual information. Parvocellular has to do more so with detail and color, and magnocellular has to do with direction of movement or motion. That's also another big idea behind this, is that when we're processing, when we're thinking about all the different features we can process from a visual image, it's usually subdivided into 2 things, which is sort of detail and color and motion and movement. And we have to have both. Sometimes, however, depending on if you're in some sort of survival context, a specific context, like I said right here, cortex and the brainstem work together to communicate to the lateral genicular gyrus to either switch from parvocellular or magnocellular primary processing. In other words, the brainstem and the cortex sometimes will tell the lateral genicular gyrus to either process its information faster because you're in some sort of survival scenario, or process slower like normal for more detail and acuity and accuracy. So you can switch between, fast, rapid fire, or slow and more detailed. Okay. I probably should have talked about these 2 things first before I talked about this. So I'm gonna have to retrace my steps a little. But talking about this is gonna make more sense about everything else we hear with the vision. So first I'll talk about neuron shape, because I guess it's more simple. So neurons come in different variety and sizes, and you can categorize them in certain ways according to certain features, function, structures, those sort of things. One way that you categorize neurons is according to their shape, and that has to do with pyramidal, stellate, and interneurons. Pyramidal, or hence the name, that shape. Stellate are more star-shaped. They also have different functions. So the pyramidal neuron is basically the main processing neuron. It has to do, this is what you have an abundance of. It is what's doing most of the processing and thinking. And it also is not very localized like stellate and interneurons. Stellate and interneurons are more local to some specific area. Pyramidal neurons can, however, connect to much farther areas. And so the idea is that pyramidal does most of the processing, most of the thinking. Stellate control where the input of information is basically going. So it controls the first part of where the input is flowing to the pyramidal. And interneurons also play a part in choosing whether to inhibit something or allow it to be excited. So they have inhibitory and or also excitatory roles. Pyramidal and stellate, however, are mostly just excitatory. Interneurons, however, are what's going to decide, should I tell the signal to stop? Should I tell it to keep going? That sort of thing. Another thing I'll talk about, okay, so this is also a pretty big idea. The vertical column. The vertical column is a word we use to describe the fact that the brain has 6 distinct layers. So most of the brain, or the inner part of the brain, if you look at it, is white matter. The outer sort of parts of it are gray. What that basically means is that the gray matter is what's the more important part, where this is where all the neuron cell bodies are located that are doing all the processing. And the white matter is just the myelinated sheath axon terminals that are going out and connecting to other neurons, either super far away or somewhere down in the brainstem into the corticospinal tract or elsewhere. The idea behind the vertical column is that you have about 200 million, I think was the number, on average, of these vertical columns, which are basically the 6 layers, and they are the individual functioning units that function to process a specific feature of vision or a specific feature of movement or a specific feature of thinking. And what they do is they communicate with each other, other processing units, other vertical columns adjacent to each other, or even communicate with others that are really far away in a sort of voting fashion by which one vertical column will receive information, think about it, talk with other adjacent or far away vertical columns in order to integrate information with them. And they basically vote and each come up with their own idea. They might inhibit something, might activate something. They each, relating to vision, each vertical column is processing a very specific part of vision, a specific angle, a specific part, specific color, that sort of thing. And so we talk about the 6 layers. So it's tempting to want to think that you go from 1 to 2 to 3 to 4 to 5 to 6 in a linear fashion. It doesn't really work that way, unfortunately. So that's what I used to think, but here's what actually happens. So at layer 4, you have the input. From 4, you go to 2 and 3 for processing. Again, with 2 and 3, you are processing, but you're also processing within yourself, and you're also comparing and contrasting with other vertical columns next to you far away. And then from there, we go to output, which is layer number 5. Now, 1 and 6 do play a pretty big role. 1 has to do with a much more executive modulating function where it modulates everything else as a whole. So you would think it goes from 4 to 2 and 3 to 5. 1 doesn't stay in any one of those parts. It's in all of them, unlike the others. And then with 6, layer 6 has to do with feedback and predictions. In other words, it will basically have a feedback loop where it basically says, oh, did what I expected happen? Like that sort of thing, in a sense, predictions. So let's get back up here. So optic nerve, optic chiasm, new system, thalamus, which is part of the thalamus, lateral genicular gyrus. Then you go to magnocellular and parvocellular. Then you go to visual area number, so 1. So like I said before, you just have to imagine that the visual area 1 is bottom, then it's 2, then 3, and then 4 and 5, and in succeeding fashion. But again, also with 4 and 5, you get the branching point. It goes to different parts of the brain, such as the parietal lobe the parietal lobe or the inferior temporal lobe. Okay, so let's talk about V1, V2, V3, V4, V5, V6, V7, and V8. So again, V1 processes the most basic features of the image. So in other words, it processes mainly lines. Like I said, high contrast lines. It's what it processes most first. So if you look at like a star, which you process first most, like say if there's like a white background and a red star, what you process first with the visual area number 1 is not so much as the color or anything specific like that, or the texture of it or anything like that. You just notice mostly the outline of it. That's the thing you notice at first. Another thing I want to point out is with each visual area, it's not like as if one of them does only color and the others can't do color too. Some of the functions that they relate to does overlap. Although some of them will be more specialized to some specific feature more than others. So let's talk, so we just talked about more about general overview. So let's talk about the specific visual areas. So visual area number 1, again, is the most basic. It processes lines, specifically high contrast lines, edges, and contrast, like I said. Visual area number 2 has to do with shapes, simple shapes, and angles. Visual number 3 has to do more with complex shapes and motion, and this is where it all branches from 4 to 5, and then from 4 to inferior temporal lobe, and 5 to parietal lobe. So let's look at 4 first, I guess. 4, like I said before, has to do with the wet pathway. It has to do specifically, it will look at more so at color and object form, and then it goes to the inferior temporal lobe to look at memories and to be able to have pattern recognition. And if you want to look at more at faces, you go to the amygdala and emotion related stuff related to the amygdala, as well as the limbic system in general. And then with 5 is the other branching point, and it has more to do with motion, direction, and speed. So again, what and where. Where, we're not talking about just where, but also where it's going to go, which has to do with motion and speed. There we go to parietal lobe. Parietal lobe, again, is where you have the somatosensory part of the brain, or one of them. Has to do with where objects are in space, as well as where they are relative to where you are. And then with 6, 7, and 8, so 6 has to do with coordinating vision and movement. 7 has to do with body orientation, specifically relative to the object in space. And again, so the distinction there is that 7 is more so just where it is relative to you, and then 6 has to do more so with coordinating vision and movement together. So in other words, using the information where it is relative to you to make more valuable information as to what the plan might be, which relates to movement. And then 8 has to do not really with processing the image. It is, and it's the last one, hence the name V8. It has more so to do with determining where the eyeball will look next. So like eye movement, using the eye muscles. Okay, so again, the general idea with movement is that you're going to take into account both higher cognitive thinking, so what your goal is. You're going to take into account also spatial somatosensory, where you are relative to the objects in your surrounding, and you're going to translate these two pieces of information into a plan. And so that's kind of what's happening here first. So the dorsolateral frontal cortex and posterior parietal cortex communicate with each other, and they each provide their own unique sort of input of what is important relative to creating the plan. Of course, the dorsolateral frontal cortex, like I showed, has to do more with higher level thinking, so the internal cues, and then the posterior parietal cortex has to do with external. And so again, this is very niche. Higher level thinking has to do with a lot of things, which is why I just wrote a few things from which could be possible contributors to internal cues. Some of which being prediction, reward, so dopamine signaling, working memory, which is like short-term memory sort of, but just a specific type of emotion. It has to do more with like limbic system. So, you think differently, you think differently whenever you're in different mood states. Goals, like I said before, this is very goal-oriented, and rules. So very complicated, not gonna talk about it. Let's move on. So dorsolateral fronticus is basically going to encode the executive goal behind the movement plan in general, whatever specific mini movement plan it generates. And then it communicates with posterior parietal cortex to, the posterior parietal cortex will basically take into account external cues such as somatosensory, vision, proprioception, olfaction, all these different sort of, your environment. To encode affordances, I'm pretty sure I talked about this earlier. Affordances is just the idea that when you're coming up with movement plans, you're not just coming up with one and then going with that one. You're actually coming up with many, and then your impulse control comes into play whenever you're actually going to communicate this plan to the basal ganglia, which inhibits most of the affordances, most of the movement plans, via the use of a neurotransmitter called GABA. And it basically, whichever one it chooses to let go through, it doesn't inhibit. And it does so by taking into account reward prediction, past outcomes, current goals. Again, that right there is more niche. I'm not going to talk about it. So the plan that it doesn't inhibit, that was created by the dorsolateral frontal cortex and the posterior parietal cortex communicating with each other, is the one that has been forwarded to the thalamus. The thalamus will amplify this signal, amplify this movement, and then it will reroute it to the PMA and the SMA. PMA stands for premotor area, and this SMA stands for supplementary motor area. The idea is that, again, like I said before, these two, the posterior parietal and the dorsofrontal, they will refine this movement plan and refine it specifically in terms of specific muscle sequences. So it translated from, I have this goal, this is my surroundings, this is what I want to do, this is how I want to move. It translates that specifically into the specific muscle sequences that have to be fired. And this can be thought in terms of temporal and spatial ways, such as, this muscle fires and this muscle fibers, and then these two are firing at the same time. And this one is inhibited, this one is relaxed. And then they are firing with X amount of intensity over X amount of time, such that, at first we fire slowly and then with less intensity, and then we do more intensity, and so on and so forth. It basically adds the muscle sequence into play. And again, kind of like I have it before, this side has to do more with sensory environment, which is why I put this on the left and this on the left, which is why, again, the PMA has to do with sensory environment, and the SMA has to do more so with not necessarily super higher level thinking, but it's something other than environment, which has to do with procedural memory and timing. So once you come up with the plan, it is rerouted to the primary motor area, and this is where everything, now up until this point, the whole plan was created. Once it goes to the primary motor area, however, this is the part where it comes into play where it will get refined some more. So it'll be tweaked a little bit, specifically by the cerebellum and the brainstem, mostly the cerebellum. But for the most part, the plan has been created. It's going to be refined, and now from this point, you're going to be looking at how you're going to carry the plan out, how you're going to execute it. And that's what the primary area does. First, before it executes it, though, by sending it through the corticospinal tract, and then from there to the upper motor neuron, to the lower motor neuron, what it actually does is it'll communicate with the cerebellum and the brainstem in a feedback-wise scenario where it talks back to basically refine it. And then once it actually is good to go, this is where the magic happens. This is where the muscle sequence movement plan is sent from the primary area through the lateral corticospinal tract, from the upper motor neuron, which is basically a way of saying that there's a neuron in the spine and the brainstem, and then the lower motor neuron is the one that basically it connects to with the upper motor neuron. It connects at whatever part of the spinal disc that is about to exit out through the ventral front side to go connect to the specific motor unit from which it connects to. And by motor movement, I mean that each neuron, depending on how fine or detailed the movement is, if it's like a broad general movement or it's some super specific movement, has its own unique motor unit, which is basically a way of saying that the neuron, one neuron can connect to multiple muscles or muscle fibers contracting, such that when the neuron fibers, all of its corresponding muscle fibers will also contract. And the idea is that for really fine, delicate movements, such as with your hands, the motor unit will have less muscle fibers per neuron, and for more general movements, such as like flexing your leg, it will have more muscle fibers per neuron. That's the motor unit. So, and then you're doing the movement. Another really final sort of idea relating to this is the fact that as you are carrying out the movement in real time, you're actually having a feedback loop sent such that information relating to what's happening as it's happening, such as you feeling your stretch in your arm, or you are seeing what's happening as a result of it, or you feel heat or something like that. Info relating to what's happening as the movement is being carried out is sent back to the cerebellum and the brainstem, specifically sent back to the feedback, the sixth layer, the vertical columns, to basically, is it going as I planned? That sort of thing. Do I need to change something? So yeah. I think that's it. Okay, so I looked back on my notes and whatnot, and I realized I was just reading through this entire thing, and I realized that there was two things that I've missed. That is Sherrington's law, which I actually wrote down, but for some reason just didn't go over, and depth perception. So look at Sherrington's law first. The idea of Sherrington's law is that with a lot of different muscle contractions, like on your limbs and whatnot, there is an antagonistic muscle to go along with it, such that whenever I, for example, contract my bicep, the triceps being the antagonistic muscle will relax. And the idea is to just produce a much more controlled movement as opposed to a jagged, like uncontrolled, shaky movement. Depth perception, I'm not too sure where I would put it on here specifically. Of course, I would probably be relating to the dark green, blue, whatever color with the vision. But regardless, instead of writing it down, I'll just talk about it. So when we think about depth perception is all about converting a 2D image, which is what you're receiving from the retina, from the eye, into a 3D image. And the importance behind that is that you use depth perception for motor movement for a lot of things. Without it, if depth perception didn't work, or if it was just more poor than it could be, what that would mean is that you would have, the world would appear flat, and you would have a lot of trouble navigating the world, being able to grasp objects or have good hand-eye coordination. And part of what that means is that, well, here's another thing. Okay, so when you think about depth perception, you can think about it in terms of two ways. You can think about it in terms of binocular depth perception and monocular, meaning depth perception arises both from one eye. So in other words, I can close one of my eyes and have the other one open. I can still have depth perception going on, but it also occurs much better when you have both eyes. You have two images coming from each eyeball. So first, and that part of the reason with that is that part of the reason why if someone is blind in one eye but they can see in the other, what this partially means is that the depth perception will probably be impaired, and as a result, again, because vision correlates to movement, they would probably have slightly harder time with hand-eye coordination, that sort of thing. So let's think about two eyes. So the big idea behind how depth perception arises specifically from having two different images is the fact that higher visual areas will compare the difference, like the disparity between, because of course there is a difference between what each eye sees. There's a disparity going on because they're in two specific places. However, most of it is somewhat the same, but there's still disparities. And so where depth perception arises is the fact that higher visual areas just essentially compare and contrast. There's like binocular neurons in the eye, or not in the eye. There's binocular neurons in the higher visual areas that will compare the image between that is being produced by both of them. The idea is that closer things, like the closer an object is, there's more of a disparity there is going to be between what is seen by both eyes, and the farther it is, there's less of a disparity you'd see. So an example, like you could practice this by like closing one eye and then opening the other, like vice versa, with looking at an object that is close to you. And you could do that also with objects that are farther, and you'll notice that the closer it is, no more disparity, and the farther it is, less disparity. So that's one of the ways by which you have depth perception. Primarily, that's the main mechanism from two eyes. From one eye, there's a few different ways. So obviously, the farther something is, the smaller it is, the closer something is, the larger it is. That's a very, very basic idea, but I mean, it's worth stating nonetheless. Another idea is that if an object is seen as blocking or occluding another object, based off of just the shape of it and its lines and whatnot, that object that is in front of it is interpreted as being closer. So that's one way. The other two ways is objects that are closer. Because it's closer, you notice more of its texture, right? So the texture is more distinct. You can see the texture. Objects that are farther away, the farther it is from you, the less of its texture that you notice, and the more whatever texture you do notice, the more it's like crowded and dense and put together and not very distinct. So yeah, texture is another way. The last one is that during head movement, closer objects shift position faster across the retina than distant ones, just because they are closer. So yeah, this is the actual end of the video.